guys, in this video, we're going to talk about the nano world. The scope of these topics are definition of terms. We're going to define terms and able to understand more. Second, areas where nanotechnology are used. And the last one is discuss what is DNA, gene therapy, and what is stem cells. But before anything else, let us know some facts about nano. The word nano is from the Greek word nanos, meaning dwarf. A nanometer or nm is a billionth of a meter or a millionth of a millimeter. 1 nanometer is about 8 times the radius of an atom and 100 times smaller than a bacterial cell. A human hair is 80,000 nanometer in diameter. Let's proceed to the definition of terms. First is nano, a combining form with a meaning very small, used in the formation of compound words which is nanoplankton. In the name of units of measure, it has the specific sense 1 billionth or 10 raised to the power of negative 9, nanomole and nanosecond. Nanotechnology, the branch of technology that deals with dimension and tolerance of less than 100 nanometers, especially the manipulation of individual atoms and molecules. Nano world of science and technology has emerged as a significant interest for governments, industries, and academicians. It refers to various technology to produce materials of extra high precision and dimension on the scale of 1 billionth of a meter. Next is area use of nanotechnology. First, energy storage. It improved capacity and safety of lithium-ion batteries decisively. Example, through new ceramic, heat-resistant, and flexible separators and high-performance electrode materials. Second, defense and security. It can be used to update existing equipment and weapons or to introduce new functions and devices. It is a broad field which can contribute to conventional as well as unconventional weapons. Next, metallurgy and materials. It can also be described as the technology of metals, the way in which science is applied to the production of metals and the engineering of metal components for use in products for manufacturers and consumers. Next is electronics. Carbon nanotubes are close to replacing silicon as a material for making smaller, faster, and more efficient microchips and devices, as well as lighter, more conductive, and stronger quantum nanowires. Next is optical engineering and communication. Technologies in the realm of nano-optics include near-field scanning optical microscopy or NSOM, photo-assisted scanning tunneling microscopy, and surface plasmon optics. Traditional microscopy makes use of diffractive elements to focus light tightly in order to increase resolution. Next is biomedical and drug delivery. Its application occurs through the use of designed nanomaterials as well as forming delivery systems from nanoscale molecules such as liposomes. Improve the ability to deliver drugs that are poorly water-soluble. Provide site-specific targeting to reduce drug accumulation within healthy tissue. Next is agriculture and food. It can increase agricultural production and its application include nanoformulation, nanosensors, and nanodevices. Nanoformulation is for applying pesticides and fertilizer for crop improvement. Nanosensor, crop protection for the identification of diseases and residues of agrochemicals. Next is cosmetics and paints. Its application in cosmetics and skincare include sunscreen that uses zinc oxide nanoparticles to block ultraviolet rays while minimizing the white coating of the skin. These proteins are encapsulated in liposome nanoparticles which merge with the membranes of skin cells to allow delivery of the proteins. Next is biotechnology. DNA is one of the important examples of bio-nanotechnology. The utilization of inherent properties of nucleic acid like DNA to create useful materials in a promising area of modern research. Next is, what is DNA? DNA, or deoxyribonucleic acid, is the hereditary material humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus, where it is called nuclear DNA. But a small amount of DNA can also be found in the mitochondria, where it is called mitochondrial DNA or mtDNA. Mitochondria are structures within cells that convert the energy from food into a form that cells can use. DNA is a double helix formed by base pair attached on a sugar phosphate backbone. The information in DNA is stored as a code made up of 4 chemical bases. A is for adenine, G is for guanine, B for cytosine, and T for thymine. DNA bases pair up with each other, A with T and C with G, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and phosphate are called a nucleotide. Nucleotides are arranged in 2 long strands that form a spiral called a double helix. The structure of the double helix is somewhat like a ladder, with the base pair forming the ladder's rungs and the sugar and phosphate molecules forming the vertical side pieces of the ladder. An important property of DNA is that it can replicate or make copies of itself. Each strand of DNA in the double helix can serve as a pattern for duplicating the sequence of bases. This is critical when cells divide because each new cell needs to have an exact copy of DNA present in the old cell. Gene therapy. Gene therapy is a form of therapy that involves inserting of one or more corrective genes that have been designed in the laboratory into the genetic material of a patient's cells to cure a genetic disease. The expression of a new gene or genes that can alter the DNA or RNA transcripts used to synthesize proteins that therefore correct the disease. Gene therapy is still the experimental stages and is used to therefore not yet widespread. Gene therapy using an adenovirus vector, a new gene is injected into adenovirus vector, which is used to introduce the modified DNA into a human cell. If the treatment is successful, the new gene will make a functional protein. The procedure of gene therapy. One of the first hurdles of gene therapy is successful introduction of the corrected gene into the human cells. Once introduced, the gene needs to be incorporated into the genetic materials of those cells. There are 2 types of gene therapy. The first one is somatic gene therapy. With this form of therapy, only somatic cells or body cells are targeted and not the germline cells, otherwise known of as the gametes or sex cells. If the modification of DNA is confined to the body cells only, then the altered genome only affects the individual treated and not any offspring produced when the gametes join to form a zygote. The other one is germline therapy. This involves incorporation of a tailored gene into the gametes, permanently altering the genes inherited by future generation. This type of therapy is prohibited in many countries due to ethical and technical concern. Next is, what is stem cell? Stem cell is an undifferentiated cell of a multicellular organism which is capable of giving rise to indefinitely more cells of the same type. Stem cells also are the foundation of every organ tissue in human body. Types of stem cell. First is embryonic stem cells. These cells are obtained from the inner mass of the blastocyst, a mainly hollow ball of cells that in the human forms 3 to 5 days after egg cells is fertilized by sperm. Tissue-specific stem cells. These cells are also referred to adult stem cells. Tissue-specific stem cells can generate different cell types for the specific organ in which they live. Next is mesenchymal stem cells. It refers to cells isolated from stroma, the connective tissue that surrounds other tissues and organs. It is capable for making bone, cartilage, and fat cells. Next is induced pluripotent stem cells. These are cells that have been engineered in the lab by converting tissue. Specific cells, such as skin cells, into cells that behave like embryonic stem cells. Next is uses of stem cell. Stem cell therapy is a treatment that uses these cells to treat the condition by generating healthy cells to replace diseased ones. Example, leukemia, lymphoma, and neuroblastoma.